Welcome to the first module of General Biochemistry. In this module, we will build a strong foundation for understanding life at the molecular level. We'll move from the history of biochemistry, through the organization of the cell, to the basic building blocks of life and how they originated. This knowledge is essential because, before you can understand complex biochemical reactions and pathways, you must understand what life is made of and how its machinery is organized. By the end of this module, you will be able to explain how biochemistry emerged as a scientific discipline. Describe the structure and organization of cells. Distinguish between different cell types and their unique features. Identify and describe the major organelles and their functions. Understand the nature and roles of biomolecules. Recognize the structural units of macromolecules and their importance in life. Appreciate theories about the origin of life from a biochemical perspective. Let's begin our journey with the historical perspectives of biochemistry. 1. Historical perspectives of biochemistry. Biochemistry, as a distinct scientific field, developed from the margin of biology and chemistry. In the early 19th century, scientists began realizing that living organisms obeyed the same chemical and physical laws as non-living matter. Before that, many believed in a concept called vitalism, the idea that a mysterious life force was required for biological processes, separate from chemistry. One of the earliest blows to vitalism came in 1828, when Friedrich Wöhler accidentally synthesized urea, an organic compound, from ammonium cyanate, an inorganic salt. This experiment demonstrated that organic molecules could be created from non-living materials, paving the way for the chemical study of life. Through the 19th and 20th centuries, scientists like Louis Pasteur, Edward Buchner, and Hans Krebs expanded the field by studying fermentation, enzymes, and metabolic cycles. The mid-20th century brought the molecular biology revolution, the discovery of the DNA double helix by James Watson and Francis Crick in 1953, which linked genetic information to biochemical processes. Today, biochemistry is central to many disciplines: medicine, agriculture, environmental science, biotechnology, and it continues to evolve with advances in genomics, proteomics, and synthetic biology. 2. The living cell, organization and molecular architecture. The cell is the fundamental unit of life. All living organisms, from the simplest bacteria to complex multicellular organisms like humans, are made up of cells. The term cell was first coined by Robert Hooke in 1665 when he observed cork tissue under a microscope. Biochemically, the cell is a highly organized compartment that carries out life processes. It maintains an internal environment distinct from the outside world, allowing controlled chemical reactions. This organization is essential. Without it, life as we know it would be impossible. Cells share some common features. A plasma membrane to control the movement of substances in and out. Cytoplasm, containing enzymes, molecules, and organelles. Genetic material that stores instructions for life. The ability to synthesize proteins and produce energy. 3. Types of cells and their characteristics. Cells are broadly classified into prokaryotic and eukaryotic types. 3.1 Prokaryotic cells. Prokaryotes are simpler in structure and are represented by bacteria and archaea. They lack a membrane-bound nucleus. Instead, their DNA is located in a nucleoid region. They also lack membrane-bound organelles, but they have ribosomes for protein synthesis. Prokaryotes are generally smaller than eukaryotic cells, often 1 to 5 micrometers in diameter. Key characteristics. Cell wall present in most species. Circular DNA molecules. Simple internal structure. Rapid growth and reproduction by binary fission. 3.2 Eukaryotic cells. Eukaryotic cells are found in plants, animals, fungi, and protists. They are more complex and larger, typically 10 to 100 micrometers in size. They have a true nucleus enclosed by a nuclear membrane, and they contain various membrane-bound organelles such as mitochondria, endoplasmic reticulum, and Golgi apparatus. Key characteristics. DNA organized into chromosomes within the nucleus. Cytoskeleton for shape and movement. Complex internal compartmentalization. Capable of specialized functions in multicellular organisms. 4. Structure, size, and functions of organelles. Let's explore the major organelles of eukaryotic cells. Nucleus, stores genetic information and coordinates activities like growth and reproduction. The nuclear envelope controls exchange between the nucleus and cytoplasm. Mitochondria, known as the powerhouses of the cell, they generate ATP through aerobic respiration. Endoplasmic reticulum, ER, the rough ER is studded with ribosomes and synthesizes proteins, while the smooth ER is involved in lipid synthesis and detoxification. Golgi apparatus modifies, sorts, and packages proteins and lipids for delivery. Lysosomes contain digestive enzymes for breaking down macromolecules, old cell parts, and pathogens. Peroxisomes break down fatty acids and detoxify harmful substances. Chloroplasts, in plant cells, carry out photosynthesis, converting light energy into chemical energy. Cytoskeleton, a network of microtubules, microfilaments, and intermediate filaments providing structural support and enabling movement. 5. Biomolecules and the origin of life. Biomolecules are chemical compounds found in living organisms, and they are essential for structure, function, and regulation. They include carbohydrates, lipids, proteins, and nucleic acids. Regarding the origin of life, one theory suggests that simple inorganic molecules in the early Earth's atmosphere, such as water vapor, methane, ammonia, and hydrogen, reacted under energy sources like lightning to form organic molecules. This was famously demonstrated by the Miller-Urey experiment in 1953, which produced amino acids from such a mixture. From there, these organic molecules may have organized into more complex structures, eventually giving rise to self-replicating systems, the earliest forms of life. 6. Structural units of macromolecules. Macromolecules are large, complex molecules essential for life. They are built from smaller subunits. 6.1 Amino acids. Amino acids are the building blocks of proteins. They contain an amino group, a carboxyl group, a hydrogen atom, and a distinctive side chain. The sequence of amino acids determines a protein's structure and function. 6.2 Monosaccharides. Monosaccharides are simple sugars like glucose, fructose, and galactose. They serve as energy sources and building blocks for more complex carbohydrates. 6.3 Glycerol and fatty acids. Glycerol is a 3-carbon molecule that forms the backbone of triglycerides. Fatty acids are long hydrocarbon chains with a carboxyl group at one end, and they can be saturated or unsaturated. 6.4 Nitrogenous bases. Nitrogenous bases are components of nucleotides, which make up nucleic acids. They include purines, adenine, guanine, and pyrimidines, cytosine, thymine, uracil. 7. Inorganic synthesis of building units. Inorganic synthesis refers to the formation of organic building blocks from inorganic precursors. Early biochemical evolution likely involved such synthesis, driven by energy from sunlight, geothermal activity, or electrical discharges. Over time, these building units combined into polymers, leading to the development of primitive metabolic systems. We've now laid the groundwork for biochemistry by exploring its history, the organization of cells, the nature of organelles, the variety of biomolecules, and the basic units that compose them. This knowledge will prepare you for Module 2, where we'll focus on proteins in detail, biochemical analytical techniques, and the chemical principles underlying life processes.